day, dear Professor Michael Sheetz. We are very honored to talk with you, to meet with you for a talk, and we want to express our gratitude for you agreeing to make time for us. And my name is Botagoz Mutaliyeva. I work at the biotechnology department of the M.Auezov South Kazakhstan University. And for me, very interesting your research work, because I know that you are a cell biologist in the Columbia University and the distinguished professor and founding director of the Microbiology Institute of the National University of the Singapore. And recently,
nanobiology.
Yes, yes, biology. And recently, as I know, 1 year ago in 2019 years, you joined to the group of the Texas, the Texas Medical Branch University as the tenured professor and in the biochemistry and the microbiology, molecular biology department. And each years ago, you was awarded by the Lasker prize. This is very, I think this is very wonderful, because this is like the same to the Nobel Prize. This as the Nobel laureate. For me, you are Nobel laureate, and so you discover some protein. Your discoveries was concerning cytoskeletal motor proteins mentioned that move cargoes within cells, contract the muscles, and enable cell movement. I mean the kinesin protein. And for our colleagues, and for me also, for the PhD students, for the master degree students, for the teachers in our university, they will be very interested in your research work. And I would like to ask you about why did you choose this direction of the research, and what the application of your results now?
Okay.
Okay.
So those experiments were done about 30 years ago. Okay.
Mm-hmm.
And at that time, we didn't understand about the motors that moved
Mm-hmm.
organelles within cells.
Mm-hmm.
And they had not been identified. And so I went on sabbatical to look into that. And quite by chance, we did experiments that allowed us to see some of these movements in the test tube. And that gave us a way to now break open cells and look for the fraction from the cell that had that motor activity. And based upon that, we then purified the motors and then identified them. And subsequently, they've been, of interest in a variety of clinical abnormalities where there are problems with the motors, etc. But indeed, it was really following up on a way to see their movements that enabled this. And that was relied upon light microscopy and some of the earliest elements of super resolution microscopy to do that.
Mm-hmm. Well, thank you very much for your answer. And also, this is very interesting from the point of the application, because I think that you discovered some mechanisms of their action of the kinesin. And so this result of this research, knowledge of this research can be influenced on the application to the medicine, for example. And what about this? What the application?
Well, the more recent work that we've done has been on mechanobiology.
Mm-hmm.
And there we are understanding how cells sense rigidity of their environment. And that is a critical element in development And also, it turns out that cancer cells are missing or do not have the ability to sense rigidity. And so we have determined that this, lack of sensing in the cancer cells causes them to behave totally different from normal cells. And we are now using mechanical means to kill those cancer cells. They are sensitive to stretch and other mechanical aspects, so that we are now applying this for let's say, augmenting treatments of cancer.
Okay. This means that this the normal movement of the kinesin and etc. And then you can observe this, and you can, for example, change something. Yes, to do more for the treatment. Okay. 8 years, and I mean the time goes on. And especially now, during pandemic of the COVID-19, we have some problems, especially I think in the development of the vaccine, etc., in the medicine. And I would like to ask you, what the maybe you change some subject, and maybe you apply your subject of the research to this direction. And I would like to ask, what the subject of research now?
Well, some of the recent work we're doing is in particular looking at what happens with senescent cells, cells which are aged, when they are exposed to virus. And the preliminary results there indicate that indeed they are more sensitive to infection, and we're trying to understand why. So it can be at a, it's clear in COVID-19 that older individuals are much more affected by the disease. And the question is that at the cellular level or at the systems level, and we have evidence that it's at the cellular level.
Yes, I think so, because COVID-19 influenced to all of the systems, and so I think the research, you should do your research in this direction, because very applicable for the treatment, and who researchers who develop the vaccines can apply your knowledge, your research results in their investigation works. And so I would like to also say about our research in our department, because we work at the biotechnology departments, and allow to introduce some of the scientific direction of our department. We have the 3 directions in our department. One of them, the agriculture biotechnology, and the second one of the food biotechnology, and the medical biotechnology. But medical biotechnology is not so this is new direction in our university, because we want to develop this direction also, but more develop it to direction, I mean the agriculture and the food biotechnology. And we work under the development of the biopreparation. I mean some of the scientists in our department work under the soil microorganisms. They investigate these soil microorganisms, isolate pure microorganisms, and then they create some of the composition of microorganisms to improve the soil fertility and the protection from the agriculture, from I mean the yes, and
so less fertilizer.
Yes, and then the second one, especially, for example, I would like to say about the chief of our department. She worked in the fermented drinks. Do you know about this? Some drink. This is national food, kumis, and she worked under kumis, and she worked under the fermentation process of this.
Okay, fermentation. Okay.
Yes. And the next one is the processing of the raw material, plant raw material. And also one of the professor investigate this fermentation processes, catalysis of the plant raw material.
Okay.
And my work, I would like to say about my own research, my research work in the field of the microencapsulation of the bioactive substances and the promoters, new promoters. I work together jointly with Ukrainian Technological State University in Dnipro and Professor Prosyanik, and also with the Croatia Zagreb University, Professor Marko Vincikovic in the field of the microencapsulation. And for me, this is very interesting then, because all of the substances need to, for example, to preserve them, to prolong their activity. And some of the plants need to have the promoters that have the more prolonged activity for their growth. I mean, this depends on the season and the time, and etc. And so we, Professor Prosyanik synthesized new promoters in amines based on the derivatives of the aminofumaric acid, and we work under the encapsulation and then testing on the plants. What difference between the microencapsulated and the non-encapsulated research, and this is our goal, and we have the projects financed by the Ministry of Education and the Sciences of the Republic of the Kazakhstan. So, but for us, I mean, our students, our university, they're very interested in your work, in your experience, because as you know, yearly I met with one professor from the Russia. He worked in the USA also, and he said that Nobel laureates can be originated from the Nobel laureates. I mean that if we met with the Nobel laureate, if we educated by the Nobel laureate, we can be also Nobel laureate. Right. And we would like to ask you to be agree, for example, to give for us your lecture sometimes, especially now in the COVID-19 period. We have some possibilities, maybe more possibilities than yearly, to meet in the online, because you haven't time to give, for example, lectures maybe face to face because of the time, exactly time, I mean the short time, and a lot of the work with the research. And I think you have the many collaboration with other scientists. All of them want to collaborate with you. But we also want to collaborate with you, and we want to that you share with us your experience. And we invite to give lecture for our students and discuss more about maybe next future and your advices. And
well, the session we had earlier was very good for that.
Okay. Yes, and especially I think for the new generation, this will be very, very great, useful. We can have the benefit from you, because I think your experience and your examples for our new generation can give for them maybe ideas and maybe some efforts and the force powers to do more and to achieve some results to
I
be
hope so.
successful. Because I know that positive people very positively influence for other people. And I would like to ask you what the advices you give for our new generation, for our PhD students, for master degree students to our university.
Well, what we discussed earlier is that you find an area that you're interested in, and your interests should take you, and you pursue that, as rapidly as you can, and get the tools that you need to answer the questions. So that's really important. Yeah. I really can't add much more from than what was said earlier, which I think is really quite important. So.
Yes, we unfortunately have so short time, but I'm very, very glad to meet with you because thank you very much for your time. Thank you very much.
Bye.